We are excited to have Nada Sheikh presenting from the Heliopolis University in Egypt. And she'll be presenting on elevated non-coding NNT AS1 relations to HSP90 in colorectal cancer patients.
Hello, everyone. Can anybody see the slides?
Yes, we can see your slides.
Okay. Good afternoon, everyone. I'm Nada Mohammed El Sheikh, a biochemistry teaching assistant at Faculty of Pharmacy, Heliopolis University. And today I'm going to discuss my master study results under title Elevated Non-Coding and NCAS1 Relation to HSP90 in Colorectal Cancer Patients, the story of negative correlation to HSA microRNA 485-5P. This study was done under the supervision of Professor Dr. Nadia Hamdi, Professor Dr. Ahmed Abu Saud, Professor Dr. Amal Fawzi, and Dr. Iman Farag Wasfi. Chapter 1, the epidemiology of colorectal cancer. Colorectal cancer is the third most prevalent and lethal malignancy worldwide, and it's the seventh in Egypt. It can be initiated through several genetic factors like hereditary mutation, sporadic mutation, or epigenomic aberrations, several lifestyle factors like obesity, lack of exercise, alcohol intake, and smoking, and several nutritional facts like red or processed meat, low intake of fibers, and high fat intake. The long non-coding RNA functions in gene regulation in the nucleus as guide, scaffolding, signaling, or decoy, and many long non-coding RNAs are exported to the cytoplasm to interact with protein or with messenger RNA for its degradation, splicing modulation, and translation inhibition, or act as competitive endogenous RNA through sponging microRNA. This is our published review in Pathology Research and Practice Journal under the title Insights on the Potential Oncogenic Impact of Long Non-Coding RNA Nicotinamide Nucleotide Transhydrogenase Antisense RNA1 in Different Cancer Types, Integrating Pathways and Clinical Outcome Association. Figure 1 represents the long non-coding RNA, microRNA, and microRNA protein possible interaction in colorectal cancer using the gene ontology classification and Coyote Encyclopedia of Genes and Genomes and Ensemble databases, which were accessed in August 2020. The long non-coding RNA NNTAS1 is located on the 5p12 human chromosome. It regulates tumor progression by sponging microRNAs, yet its role in colorectal cancer needs further investigations. The Homo sapiens microRNA 485-5P is located on the human chromosome 14q32. It acts as tumor suppressor gene in several human cancers through regulating the expression of several downstream genes. It's studied by our group in breast cancer by Dr. Marwa Mahmoud, who won the Top Presenter Award in BSI 2021. But its regulatory role in colorectal cancer is not explored yet. Chapter 2, the materials, aim, and method of our study. Our study was done. The aim of our study was to study the molecular signature of NNTAS1 and its downstream gene microRNA 485-5P and HSP90 in colorectal cancer patients' liquid biopsy, and to highlight the NNTAS1 microRNA 485 relative expression using quantitative real-time PCR and HSP90 protein concentration using ELISA as a possible diagnostic molecular markers for colorectal cancer. Chapter 3, the subjects. G-power sample size online calculator was used to estimate the sample size of the study. The control samples were obtained from apparently healthy volunteers, and the colorectal cancer patients were recruited from the National Cancer Institute after signing an ethically and approved informed consent. The number of samples obtained from the control were 28 in ratio 13 male to 15 female, and the number of samples obtained from the colorectal cancer patients were 60 in ratio 30 male to 30 female. 13 of them were grade 1, 28 of them were grade 2, and 19 of them were grade 3. Our inclusion criteria was histologically confirmed primary colorectal cancer adult patients, and exclusion criteria was patients receiving a chemotherapeutic treatment, radiation, or undergone surgery, patients suffering from any cancer other than colorectal cancer, patients with incomplete data or histopathological diagnosis, or patients with metastatic colon cancer. The demographic data recorded from patient profiles were family history, smoking status, liver and kidney functions, and hematological profile. And the clinical pathological data were tumor site and size, mucinous or not, TNM, vascular invasion, tumor differentiation, staging, and inflammation status as inflammatory bowel disease. And the ratios and index calculated for all the participants were body mass index in kilogram per meter square and the platelets to lymphocytes ratio. To investigate the functional enrichment of genes and pathways, the following databases were accessed in August 2020. The gene set enrichment analysis was cluster profiler using the gene ontology classification and Kyoto Encyclopedia of Genes and Genomes, Ensemble, HGNT, Genome Data Viewer, NCBI, and store-based database. Results: the serum expression pattern of NNTAS1 was significantly increased in colorectal cancer patients in comparison with control, as well as each grade versus the control. Our study results goes in compliance with Wang et al., who reported that upregulation of long non-coding RNA NNTAS1 leads to colorectal cancer cell migration, proliferation in vivo and in vitro. The serum samples of the subjects, as we can see, the microRNA 485 was significantly upregulated in colorectal cancer patients in comparison with control, as well as each grade versus the control. Our study results goes in compliance with Jiang et al., Hui et al., and Cheng et al., who reported that microRNA 485 expression level was downregulated in glioma, ovarian, and lung cancer. The serum concentration of HSP90 was significantly increased in colorectal cancer patients in comparison with control, as well as each grade versus the control. Our study results goes in compliance with Dang et al., who reported that high HSP90 levels are associated with poor prognosis in colorectal cancer patients. And our study revealed that by combining NNTAS1, microRNA 485, and HSP90 to CEA and CE199, there was an improvement in the diagnostic utility of the current diagnostic markers. Our study results goes in compliance with the STAR-based database, which revealed that there is an interaction between NNTAS1 and microRNA 485, as well as microRNA 485 and HSP90 AB1 gene. In addition, there was a significant negative correlation between NNTAS1 and microRNA 485, as well as microRNA 485 and HSP90. However, there was a significant positive correlation between NNTAS1 and HSP90. The correlation analysis comes in line with the bioinformatics results and suggests that the NNTAS1, microRNA 485, and HSP90 work together in favor of colorectal cancer development. To the best of our knowledge, this is the first study to postulate the correlation between NNTAS1, microRNA 485, and HSP90, which may act as a probable axis to determine the incidence and progression of colorectal cancer. The story last chapter, Future Perspective for Happily Ever After Ending and Better Health, considering microRNA 485 5P as a potential precision therapeutic target for colorectal cancer treatment based on histologic grade subclassification by repurposing drugs by molecular docking to target microRNA 485 5P downstream genes obtained by MIRDP and after proving the mechanism in vitro experimentally. Thank you so much.
Great, thank you so much, Nada. Now I will open up the floor for any questions.
Sure, I have a question. That was a great presentation. Thank you very much. Very informative, Nada. My question to you is, when you showed the HSP90 levels, certainly they were elevated in all stages of the cancer that you showed there. So my question to you is it really prognostic of progression, or is it just a marker for the presence of cancer?
This is a great question. No, it's just a mark for colorectal cancer. We didn't make the overall survival rate for the patients.
Thank you. I'll jump in with another question then. So you mentioned targeting miRNAs. Have you had any thoughts how you might do that?
I'm sorry, can you repeat again the question?
You mentioned the link between the miRNA that you identified and HSP90, the potential to regulate that. How, what are your thoughts in terms of targeting? Would you actually target the miRNA or would you target HSP90? Where are you going with that? What would your strategy be?
We want to go with personalized drug therapy through targeting the HSP90, targeting the genes of the microRNA through gene-gene interaction. We can block it using several drugs like acetylsalicylic acid or other drugs.
Thank you.